Hi everyone. Today we are going to talk about the structure of the plasma membrane. Uh, every cell has plasma membranes, bacterial cells, plant cells, animal cells, but there are minute differences in between the plasma membranes of species. Here we are going to talk about a typical plasma membrane, especially animal plasma membrane. You know, plasma membrane isolate a cell from its external environment. In animal, we call this external environment as extrastitial fluid. Extrastitial fluid. Inside this plasma membrane, you have the cell plasma. It is mainly composed of water, ions, and other molecules. In this plasma, cell organelles are floating and swimming inside. If we come to the molecular structure of the plasma membrane, you can see these blue color molecules. Each molecule has a head and two tails. We call these molecules as phospholipids. Phospholipids. Most commonest phospholipid in animal cells is phosphatidylcholine. Phosphatidylcholine. Here you can see these phospholipids are arranged in two layers. This is one layer, this is the other one. If we take this side as the outside the cell and this side, this area is the inside of the cell, the external layer facing outside and internal layer is facing inside. In between these two layers, you can see these red color molecules are embedded in. These are proteins. Some proteins penetrate the both layers of the plasma membrane. Some proteins penetrate only one. And some proteins don't penetrate the lipid bilayer. They are only lying on the surface of the phospholipids. Right? So you can understand there are various kinds of proteins in the plasma membrane. In between the two layers or the bilayer, bi means two, in between the two layers of phospholipids, you can see these green color molecules. These are cholesterol. They are lying in between the two layers of phospholipids. Okay. Here you can see these black color molecules. They are carbohydrates. They are carbohydrate groups. They are attached to both phospholipids and proteins. When they are attached to phospholipids, we call them glycolipids. Glycolipids. When they are attached to proteins, we call them glycoproteins. Right? Now you can see this chain like molecule. This is also a protein, right? This is attached to this surface protein. This chain like molecule is a part of the cytoskeleton. This is a part of the cytoskeleton. We are going to talk about the cytoskeleton in upcoming videos. What is a cytoskeleton? Cytoskeleton is a complex network of proteins inside the cell plasma. This cytoskeleton helps to cell trafficking. Inside the cell, they helps to move organelles here and there, and they helps to move the plasma membrane and change its shape. The cytoskeleton helps in various functions of the cell. We are going to talk about that in the future. Okay. Now, if we take a simplified molecular structure of the phospholipids, this is a simplified version of the phospholipids. Here you can see a phospholipid molecule has various components. Here the chain like, string like two tails are fatty acid chains. Fatty acid chains. Those two fatty acid chains are attached to this molecule. This part is a glycerol molecule. Glycerol. This is a 3-carbon molecule. Here there is a phosphate group. There is a phosphate group. There is a phosphate group attached to this glycerol molecule. Attached to the phosphate group, sometimes there is a choline group. In phosphatidyl choline, the commonest phospholipid in animal cells, you have this choline molecule. Okay. If we take the chemical structure of these components, we call these parts, these three parts, as head of the phospholipid molecule. These two fatty acid chains are the tail of the phospholipid molecules. But these two groups, head and the tails, are different in chemical nature. The head is a polar. Head is a polar group. Polar means this group's electron cloud has a charge. The electron cloud of this group is charged. So they can make interactions with another polar molecules like water. So this group can make, head area can make interaction with water molecules like this. They like water. They make interaction. This area can dissolve in water. So we call this head group hydrophilic. Hydrophilic head. What is the meaning of hydrophilic? Hydro means water. Philic means like. This head area likes water. It dissolves in water. But the tail is the opposite. Tail is hydrophobic. Hydrophobic. Tail is hydrophobic because it is non-polar. Non-polar. Non-polar means the electron cloud of this area is not charged. So it cannot make interactions with water because water is a charge or polar molecule. Because so this area doesn't like water. It cannot dissolve in water. That's why these phospholipid molecules hide their fatty acid chains themselves. In the extracellular compartment or interstitial fluid, you have plenty of water. Also in the intracellular compartment, you have plenty of water. That's why polar head or hydrophilic head is in contact with water in the outer membrane, also in the outer layer, also in the outer layer and also in the inner layer. The charge head is making interactions and in the close contact with the water. But their hydrophobic fatty acid chains hide themselves in between the two layers. They can't make interactions with water. So they hide themselves in the lipid bilayer. Okay. Then we come to the structure of the cholesterol molecule. In the cholesterol molecule, you have this polar head. And this black color area is made out of steroid carbon chains or carbon cycles. This is a steroid carbon cycle. And here you have the hydrocarbon chain. Right. Now let's see how these cholesterol and phospholipid molecules are arranged in the plasma membrane. Here you have the phospholipid molecules. The polar head of the cholesterol molecules is having a close contact with the head of the phospholipid molecule. Right? Here it is the cholesterol molecule. Here it is the phospholipid molecule. The head of the phospholipid molecule is polar, and also the head of the cholesterol molecule is also polar. Both of them are hydrophilic. They like water and they like each other. They can make interactions. Polar molecules can make interactions. The two heads make interactions, and this area of the fatty acid chain of the phospholipid molecule is nonpolar, and these steroid carbon cycles are also nonpolar. So nonpolar molecules can make interactions each other. So the fatty acid chain and the steroid carbon cycles make different types of interactions between them. When there is cholesterol in the plasma membrane, due to these interactions, these fatty acid chains become less mobile. Because these fatty acid chains are making contacts or connections with the cholesterol molecules, they become less mobile. So when there is cholesterol in the plasma membrane, the plasma membrane become less permeable. Now you can understand cholesterol can reduce the permeability of the plasma membrane. But the special feature of cholesterol is it doesn't reduce the fluidity. What is fluidity? The two lipid bilayers is not a rigid structure. They act like a fluid. They can change its shape. So the plasma membrane is not a rigid structure. It is flexible fluid structure. When cholesterol is in the plasma membrane, they make connections with the phospholipids and reduce its permeability. Okay. Now we come to the fluidity of the plasma membrane. Actually, phospholipids are the molecules which gives the plasma membrane the fluidity. Right? They make the plasma membrane dynamic, movable. They helps the plasma membrane to act as a fluid. Okay. What are the factors that affect the fluidity of the plasma membrane? First one is the temperature. First one is the temperature. You know, when the temperature increases, the motion of the molecules in any molecules, the motion of the molecules increases, speed of the molecules increases. Then fluidity of a sample is increased. Even in the plasma membrane, when the environmental temperature increases, the fluidity of the plasma membrane increases, right? Then the second factor is the chemical nature of the phospholipids. Chemical nature of the phospholipids. When these fatty acid chains of phospholipids become short, right? You have to understand there are various types of phospholipids. There are various types of fatty acid chains combined with different types of glycerols, cholines, phosphate groups. So, when combining, they can make various types of combinations and make thousand types of phospholipids. When the length of the fatty acid chain reduces, it makes less connections with adjacent phospholipids. When the length of the fatty acid chain reduces, it makes less connections with less interactions with the adjacent phospholipid molecules. Why is that? When the length of the fatty acid chain reduces, the electron cloud of this area reduces. So, with less electrons, they can only make few interactions. So, interactions between phospholipids reduces, then movability of these phospholipids increases, and they become more fluid. Their fluidity is increased. When the length of the fatty acid chains reduces, fluidity of the plasma membrane increases. It is one chemical factor of the phospholipids which affect the fluidity of the plasma membrane. The other one is this type of kinked fatty acid chains. Kinked. We call this is a kinked fatty acid chain. What is a kinked fatty acid chain? In this straight fatty acid chain, all the carbon molecules are bound by single bonds. So they have made the maximum number of the chemical bonds they can make, right? Because they have made only single bonds, they have made the maximum number they can make as atoms. So in a straight fatty acid chain, each and every carbon molecule has make 4 bonds. It is the maximum number of bonds they can make. We call them saturated fatty acids. But these kinked fatty acid chains, they make double bonds. their carbon molecules make double bonds. CH2, CH, double bond, CH, CH3. Because of this double bond, these carbon atoms are cis. We call this bond cis bond. It is a double bond. We call this as a cis bond. Because of this cis bond, there is a bend in the carbon hydrocarbon chain. There is a bend in the hydrocarbon chain. We call these kinked fatty acids or fatty acids with double bonds. We call them unsaturated. Unsaturated fatty acids. Unsaturated fatty acids. Because of this kink, the space between phospholipids are increased. Space between phospholipids are increased. So, these phospholipids with kinked fatty acids are very hard to freeze. So, they can stay as a fluid in lower temperature than these phospholipids with a straight or saturated fatty acids. In bacteria and fungi, when the temperature reduces, when the environmental temperature reduces, they increase the synthesis of unsaturated fatty acids. They increase the synthesis of unsaturated fatty acids. By that, they increases the synthesis of phospholipids with unsaturated fatty acids. They use these phospholipids to build their plasma membrane. So, in lower temperatures, their phospholipids become very hard to freeze. So, they can live in lower temperatures without dying because of these unsaturated fatty acids. If we come to archibacterial plasma membrane structure, here you can see the structure is slightly different because most of the time, instead of fatty acids, they contain prenyl molecules instead of fatty acids. These prenyl molecules are 20 to 25 carbon long, so they are very long molecules, and they span from one monolayer of the plasma membrane to the other monolayer of the plasma membrane. So, there are two layers of phospholipids are interconnected chemically via these prenyl molecules. So, these archibacteria can withstand very high temperatures. In other organisms, when the temperature increases, these phospholipids fluidity increases and plasma membrane structure is destroyed. But in archibacteria, in high temperatures, the structure of the plasma membrane is maintained using these special type of phospholipid molecules. That's why archibacteria can live in hot water springs and reaching up to 100 or more temperature. In the next tutorial, we will be talking about the function of the plasma membrane in detail. In that video, we will talk about the cell membrane transportation, how molecules get into the cell and get out of the cell through the plasma membrane. Okay, thank you everyone.